probably about reaching your saturation of science stories, so I'll try to be brief. First of all, I just wanted to thank all of you for your support. It's highly motivating for us to be here and to see so many people that are supporting us, and obviously it's very enabling as well. So I'm a cell biologist, and so I'm like a cell doctor. I don't take care of cancer patients, I take care of cancer cells. And specific things that we do is related to some of the things you've heard, but the general principle is that when a pathologist first looks at a section from a tumor, he sees a lot of things that have gone wrong. The nucleus is forked and misshapen, the cells have an odd morphology, the cells are invasive. And when a geneticist does a molecular dissection, he finds, as Mark said, that there are many alterations in many genes. And so part of the challenge is figuring out which of those genes are actually driving the specific events that are involved in initiating tumorigenesis and in causing a progression of tumorigenesis. So as a cell biologist, we've built a cell culture system in which we can culture normal breast cells under conditions in which they can form little hollow spheres, which look very much like each of the little glandular units of your breast. So your breasts are kind of formed of grape-like clusters. Each of the little units produces milk when it's appropriate time. So in culture, the breast cells can form such structures, and then we can introduce the genes which are identified by geneticists as being altered and say which of these genes are able to cause abnormal changes in the behavior of the cells, and then we can specifically define which events are caused by this. So we can see which genes converts a non-invasive lesion into an invasive lesion. We can see which ones allow the cells to proliferate uncontrollably. And we've specifically focused on genes, or one of the focuses on genes that allow cells to survive when they're outside of their normal homes, so to speak. So every cell is in a specific niche, and normal cells, when they move away from the niche, undergo cell death. The cell eliminates those cells. There's very strong controls to make sure those cells don't proliferate. Well, the altered genes that are associated with cancer allow those cells to survive when they're outside their homes. So we're establishing the mechanisms that are responsible for that, and then working with clinicians and pharmacologists in order to develop drugs that will prevent those cells from surviving outside their niche. And we have several genes and several drugs that we're working on now. Hopefully someday they'll have some impact for people with breast cancer.